With this figure, we want to talk about what are called chemical synapses. And what we're going to refer to here is this image at the top where we're focusing on where a neuron synapses abuts against a target cell. And that can be either another neuron or it could be a target cell of some type. And with chemical synapses, there's actually more than one type of synapse. Chemical synapses are defined by their release of a neurotransmitter. Okay, so let's back up though for a second. If our presynaptic cell, this neuron here, is experiencing a nerve impulse, right? We've got these repeated action potentials being generated along the axonal membrane. What changes the difference at the synaptic membrane is the presence of these voltage-gated calcium channels, right? So now we reach the threshold of a new type of channel. And because calcium is found in such high concentration outside the cell, it will enter the cell when we open these channels. That calcium is a stimulus for vesicles to fuse and exocytose their neurotransmitter content. Now, these vesicles are actually produced upstream in the neuron and travel along the cytoskeleton using little molecular motors to accumulate down here at the synaptic bulb. So we have a huge number of these vesicles, each one containing thousands and thousands of molecules of neurotransmitter. So every time a nerve impulse arrives, we get a few of these vesicles synapsing and releasing their neurotransmitter. Not all of them, just a small number. That neurotransmitter will diffuse across this space, which is known as the synaptic cleft. It is a very small distance, and that tends to minimize the time required for that diffusion event across that space, 20 billionths of a meter. And then on the other side here, on our target cell, we're going to have receptors. And those receptors can be found on what are called ionotropic channels, or they can be found associated with a separate mechanism. But let's focus on those ionotropic channels for just a minute. What we see is that binding of our neurotransmitter opens this channel with which it's associated, right? It's essentially part of that channel. And so in response, we would get ion flux. And depending upon which ion is moving, which ion we have permeability to, we would get either an EPSP or an IPSP on this postsynaptic cell. And of course, that would lead to electrotonic current. Now, the other option is that our neurotransmitter can bind to a receptor that is not associated with a channel. Instead, it's associated with these transduction proteins. And they're sort of like middlemen. What they're going to do is they're going to transduce this receptor binding into production of this thing called a second messenger molecule. Now, that second messenger molecule can go on and bind to a receptor and open a channel. It's sort of like a delayed ionic flux, right? It's still a metabotropic channel because it's requiring opening via the second messenger molecule, but we end up with sort of the same end result. Now, an alternative, or in addition even, effect of this second messenger molecule is that it can cause other non-channel cellular effects. It can change the metabolism, the functioning of this target cell in ways other than simply altering its membrane voltage. Now, one of the last thing to point out here is that our neurotransmitter is very short-lived due to the expression of these enzymes, which are often found in this postsynaptic membrane. And so these enzymes chew up and degrade that neurotransmitter, sort of shutting off our response. And what that allows is that our arriving action potentials here, nerve impulses, at our presynaptic cell will generate individual events on our postsynaptic cell. It's hard to get them to sort of blend together, except at very high action potential rates in our presynaptic cell. And again, because our neurotransmitter is being degraded so rapidly.